Welcome to the Endpoints podcast, presented by the ALS Therapy Development Institute. I'm Jonathan Gang. When you're trying to develop a drug to treat or cure a disease, it can be really helpful to know what specific biological properties you want it to affect in order to produce your desired outcomes. These properties are known as drug targets. Today we're joined by ALS TDI's Chief Scientific Officer, Fernando Vieira, to help us understand what a drug target is, how they're identified, and what we're doing to try and find effective drug targets for ALS. So when we talk about a drug target or a biological target, what are we talking about? What does that mean?
So a drug target is, it's a target, it has a biological function that's specifically implicated in human health and or disease. So drug targets can be genes or they can be proteins that do an important job. When you're thinking in the context of a disease, you know that they're playing a role in driving the disease processes or the disease symptoms.
And what is the role of a drug target when we talk about drug discovery?
So a drug target, it plays a function of being something that you can try to modulate. You can either try to block it, or you can try to activate it. You can try to reduce it, or you can try to increase its levels so that it will influence disease progression or symptom progression. You want to influence it in a way that makes either the disease slow down or resolve or ameliorate symptoms.
So we're looking for something to change with a drug and then looking for drugs that will change that.
Yeah, so I guess it's worth saying that a lot of drugs get discovered without having a target, right? So a lot of drugs that have been identified through time have been discovered using processes called phenotypic discovery. So that is to say, a person has a disease and a doctor tries lots of different things, lots of different molecules, medicines, therapeutic approaches, and they get better. And then somebody tries to figure out after the fact how they got better. And sometimes that process is what reveals a drug target. And then so you figure out, well, what was that drug doing in the cell? What was that drug doing in the organ of interest? They find a specific protein that might have been inactivated or inhibited in some way. And then that becomes the drug target against which you can create or discover new molecules that even do the job better. So you don't always need a drug target to find an efficacious drug, but once you have a good drug target that's validated, it's a shortcut that gets you straight to better drugs that can more effectively treat disease.
Are there any examples of well-known targets for other diseases that people might have heard of?
Yeah, so in like HIV is a disease that I think people are aware of. So HIV is a virus that replicates inside the cell, and the replication process requires reverse transcriptases. They're enzymes inside the cell. So there are a class of drugs called reverse transcriptase inhibitors that are really, really good at stopping HIV from replicating inside a human cell, blowing that cell up, and then spreading throughout the whole body. So that's a pretty good example of a good drug target.
And what is the process of identifying or discovering a drug target for a particular disease usually entail?
There are lots of different ways you can identify a drug target. Like I described a little bit earlier, one of the ways that drug targets are discovered is by looking at a treatment or a drug that already works for a disease and then analyzing the tissues and cells in the context of treating the disease to see what's changing, to see if there's a protein that's changing, or if there's something that's levels go up or down, or it stops functioning. That's one way to discover a drug target. More recently, in the last couple of decades, whole genome sequencing and genome association studies have gone a long way to identifying drug targets. So you'll analyze the genomes of people who have a given disease, find genes which are mutated often in those people with a disease, and that gene encodes a protein which can often be a drug target. I say often, but not always. Not all genes that are associated with a disease make good drug targets.
Could we talk a little more about what makes a good drug target?
Sure. So a lot of good drug targets they're proteins, and historically, good drug targets have been enzymes, ion channels, transporters, and receptors. So enzymes are proteins that catalyze a reaction in a cell. They do a job that changes something else in the cell, and they can be inhibited. Ion channels usually are on membrane surfaces of cells, so either the cell membrane or the membrane of a mitochondria or a nucleus or something like that, and they allow sort of sodium, potassium, calcium, all of these important charged ions to get into and out of the cell, which regulates behaviors of those cells. And then transporters aren't limited to ions, but they might do a job of importing a specific protein from outside of the cell and taking it into the cell so it can do a job. That's another type of thing that can be inhibited. And then receptors are cell surface. You can imagine them almost as like a lock. They're cell surface locks that keys will kind of associate with them and activate entire cascades of processes inside of the cells. So these are things that have typically served as good drug targets. But more generally, a good drug target is something that has confirmed function within your disease of interest, right? So you know that it is changing or it is involved in the processes that are causing your disease. That target should be something that isn't expressed on every single cell in your body. It should be something that's more specific to the part of your body that is involved in the disease. Altering this target should not cause, in a normal setting, in a healthy person, if you gave them this drug, it shouldn't inhibit processes that are really important to normal functioning. So it should have almost no effects on someone who's healthy. And then another key element would be if you identify a target, it should be something that you can build an experimental system in the lab that allows you to find better drugs that influence that target. So those are criteria that make for a great target.
So once you have a target that you at least think might be good, how do you use that information in the lab? How do you build experiments around it?
That's a great question. So say one identifies an enzyme that is overactive in the context of a disease, and you believe it makes sense to block that enzyme's activity. You can build a series of tools in a lab that allow for you to identify ways to block it. So the most basic system is to manufacture a lot of that enzyme, put it into a solution in the equivalent of a tube, give the enzyme the components that it would typically act on, which might be another protein that's called a substrate. And then you measure under normal conditions how much of that substrate is changed by the enzyme. If you want to have an inhibitor, now you make a whole bunch of those tubes filled with a whole bunch of that protein and substrate, and then you test lots of different molecules that you anticipate could block that reaction. And then you measure that change in the reaction. The molecules that do a good job of reducing that reaction rate end up being called hits. And then you can now take those hits and you kind of move to a more complex system. Maybe now you're working, instead of just the protein in liquid in a tube, now you're moving to cells that are expressing the protein. And you want to see if your molecule can inhibit that protein in the context of a living cell. And you measure that output. And at this moment, now you're starting to notice if these molecules also kill the cells, right? So do they do things they're not supposed to? Does inhibiting this target actually kill the cells? Sometimes you don't want them to do that. In the case of cancer, sometimes you do. And then once you find molecules that do a good job of hitting that target in cells, then you might take them up into an animal model that also expresses the target, theoretically in the context of disease. So you want the animal model to recapitulate elements of the disease you're studying, and you see if inhibiting that drug in the context of disease in an animal makes the animal's disease severity get better. Is it ameliorated? So that's kind of the process. Having that drug target gives you that linchpin where you can build processes to reveal the best possible drugs moving into human clinical trial.
And to that point, we talk about what makes a good drug target, but what really makes something a bad drug target?
Yeah. So it's kind of the corollary of what makes things a good target. A bad drug target is a sort of fundamental control switch to lots of biology that allows us to stay alive, right? So if you are trying to, I'm trying to think of a good analogy for like a home, right? So if you have a leaky faucet, you can get rid of the leak by turning off the water main. And that's, that the equivalent of that in biology would make for a bad drug target. You really want to get more precise with your approach to solving the problem.
And then let's talk about ALS now. What are some drug targets that we have to work with in ALS?
There are a few that have been looked at in ALS, and we've talked a little bit about it during this, but one good target for ALS, at least some cases of familial ALS, is clearly SOD1. So superoxide dismutase is protein encoded by a gene which we know is mutated in ALS, in some ALS cases. And there's a lot of evidence suggesting that reducing levels of the misfolded protein could be therapeutic. And that's been studied by lots of different groups, including ours, but many, many groups in the ALS space have looked at that. And in fact, there's an interesting approach looking at antisense oligonucleotides reducing SOD1 levels in familial SOD1 ALS being pursued by Biogen right now. So that's one really good target. Other interesting targets include cell surface receptors that are important for the activation or dysregulation of the immune system. There's reason to believe that the immune system in ALS, not just familial ALS, but also sporadic ALS, is dysregulated. So if you block activation, of certain inflammatory processes by inhibiting specific receptors. There's one that ALS TDI has looked at called CD40 and CD40 ligand, blocking that pathway. But others as well. I know CD14 is a target being looked at right now in the clinic. These are interesting targets for ALS as well. And then there's been a considerable amount of effort looking at targets that might influence the ability of cells to properly fold proteins that are predisposed to misfold and wreak havoc. So influencing things like protein chaperones that help orient proteins into their proper conformation. So, but the list of interesting targets in the ALS space is really, really long, but they're not, many of them don't fit the perfect criteria for a good drug target. A lot of the genes which have been implicated in ALS have a lot of fundamental roles in a lot of biology, so it makes them tricky to modulate without doing harm. So we continue to strive to identify the best targets while acknowledging that we may need to delve into more challenging target spaces for ALS, which is clearly a very challenging disease.
Yeah, and then can you maybe expand a little bit on just how are we searching for more drug targets for ALS? What kind of experiments are you looking at to sort of identify new things to focus on?
That's a great question. So there are lots of different ways to try to evaluate targets. And at ALS TDI, we're executing lots of different types of experiments to do that. First of all, we do lean on the broader research community. We read the literature. We look at other people's whole genome sequencing studies, genome association studies. We look at results of therapeutics that may seem to be helping in specific contexts. So we definitely kind of keep our eyes open for what's out there. Internally, though, we conduct a lot of what we would call phenotype screens or phenotypic screens, where we generate systems that either in cells or in animals, and we study systems in cells or in animals that develop elements of ALS. So cells that die because they express a protein that's known to be implicated in ALS, or mice or fish that get sick because they express a protein. And we look for therapeutics that influence them and make those systems have ameliorated disease. Then when we see an improvement, we will ask targeted questions, pun intended, specific questions about what biological processes are changing when we get better outcomes. Those biological processes that change in correlation with better outcomes reveal targets. They reveal the things that you want to try to modulate better to improve the outcomes even more. Sometimes it turns out that you can't improve on it because there are lots of redundant processes, and when you push down on lever A, lever B compensates for it. But every now and again, you do come across something that stands out, or it might be an enzyme or a receptor that stands out as something that you can specifically impact to get a good outcome. And we hope that those can end up leading us to good therapeutics for ALS.
Dr. Vieira and his team at ALS TDI are continuing to work to find new potential drug targets for ALS. Thanks for listening. If you have questions, comments, or feedback, please email us at endpoints@als.net.